Okay, my presentation, I have added some slides after having given the text. So there may be a shift between the numbering in the document and the numbering of the slide. You can find the numbering of the slide somewhere here, but there can be a slight shift. So my presentation will be in 4 parts. First, I will present you the Montpellier research area and the different subjects that are developed, the different institutions which are present in Montpellier. Then I will present you the Faculty of Agronomy of Montpellier-Supagro, which is my university, and the possibility of exchanges between my university and yours or other universities in the world. Then I will present you the research department in plant physiology where I am working. And finally, I will give you 2 examples of research projects that are developed in my group. This might be maybe a bit technique or a bit complicated to understand, but I will try to make it as simple as possible. So first, Montpellier is in France. You know, in the south of France, Paris is somewhere here. So Montpellier is close to the Mediterranean Sea. Okay, it's not a very old city. It's only 1,000 years old. It's close to the sea, but it's not exactly at the sea. And it's an agricultural region, mainly, and tourism. There is a lot of agriculture and tourism. So it's hot compared to France, but not so hot as here. And we have a lot of production of wine in this region, so we have a lot of teaching in wine and enology. The population of the city is around 300 and 400,000 people, depending whether it is only the city or the city with the suburbs. And we have a lot of students, because you see that we have 70,000 students, so it's a very young city, attracting students from a lot of places. So Montpellier hosts the highest scientific concentration in Europe in the fields of agriculture, food, biodiversity, and the environment. We have 2,700 researchers and professors, 5,000 students, and this makes overall 10,000 people working in this field of expertise. So this is quite a huge place at the world level. A lot of scientific disciplines are developed in this area. It covers agronomy, ecology, biology, genetics, etc., environmental sciences, and also social sciences. So it integrates both technical aspects and more social and economic aspects. And there is a special place for mathematics and computing at all steps, for modeling, for giving spatial information to monitor the area and to monitor the production, biostatistics, and also robotics to automatize the work which is hard in the field, you know. So all this federation is under the banner of Agropolis International. This is the name of this federation. And this is the headquarters of Agropolis. It's only a federation, you know. It's important for communication, for giving information from Montpellier to the world. So we are well known in the world because of Agropolis, which puts everybody in order of work. And the other interest of Agropolis is to make people work with each other also, to facilitate the collaboration between all the different disciplines that I showed you, to favor the connection between the different disciplines by 2, by 3, etc., to make a powerful research development. So it gathers quite a lot of institutions, 27 higher education and research organizations. Universities, the University of Montpellier, 2 universities of Montpellier actually, these 2, and also from cities which are nearby Montpellier. 10 engineer schools, and I am belonging to Montpellier Supagro, which is one of the engineer schools. Some of these engineer schools are in agronomy, some of these engineer schools are in environment, and the others engineer schools are more on the economic aspects and marketing, economy, etc. Also, we have 13 research institutes, and you may know the very famous CNRS, which is the leading, one of the leading in the world research centers, and INRA, which is more applied science, but which is more devoted to agriculture, agronomy, in a broad sense, and others. And we have also international members of this Agropolis community through USDA from USA, CSIRO from Australia, and we have also permanent people from Embrapa, Brazil, who are located in Montpellier. So, Agropolis hosts the European LabEx of Embrapa. So Embrapa has different laboratories in different places in the world, and one of these laboratories is located for Europe in Montpellier. And this is since 2002. So we have on a regular basis between 2 and 4 researchers from Embrapa, who are present in Montpellier. And there are also regularly calls for shared projects between researchers from Montpellier and researchers, Brazilian researchers. Presently, the topic which is favored by the LabEx of Embrapa is about agri-food technologies, plant physiology, and development of advanced methods for phenotyping of plants in the field, you know. So, Agropolis addresses major societal challenges. So, we work on sustainability, develop the sustainability of the agriculture and agronomy. To improve, the objective is to improve food security and nutrition, good nutrition. The idea also is to take care of the natural environment. Also to go forward towards an ecological transition. And finally, to prevent damages to the environment. These are the main directions that are supported and put in front by Agropolis, and all the researchers working in this field are supported to develop this kind of research. So it is also a pole of scientific excellence. It is not really applied research, it is more basic research. And as you may know, the privileged way to evaluate the basic research is through publications and the quality of the publications. My colleague from University of Lavras already told you about that before. And here, according to this criterion, the poll is of a very good level. Of course, we could criticize this way of evaluating the research. We could consider that it would be better that the research is transferred and to evaluate the efficiency to transfer the information from the research to the field. But this is it. This is how research is evaluated worldwide and also in France, and there is a lot of pressure for that. And you saw my colleague from the University of Lavras speaking of ranking of the universities, and all this ranking is about the quality of research. So we have to be good and to develop good research. And so the quality of the science developed in the Agropolis International group is very good. There is not only research, there is connection between research and teaching, and a lot of researchers are involved in the teaching, bring the new information, the new technologies that they are studying in the teaching. So there is a tight relationship between the two. We have more than 150 degree courses for 5,000 students at all levels, 6 graduate schools, thematic graduate schools on different topics. And what we would like to develop more is the vocational training, that is continuing education for professionals. This is not very developed in France. In France you do your studies and then you go to work, and it is it's not often that people come back from the work to go on their studies, and we would like to develop more this aspect. And also e-learning, which we try to develop more and more to be able to teach to people who are not located in Montpellier and who are located in other countries, in partner countries. It's a platform with an international scope. I already spoke that to you, detailed that to you. All these organisms, CIRAD, IRD, as the French institution which are devoted to cooperate with southern countries and support them. We also are in connection with the institutions from different countries, and I already mentioned Embrapa and Brazil. And we recently welcomed the CGIAR headquarters, which is the head of the CGIAR, and it is now located in Montpellier. And we are partner of different international networks. So we also have a strong international partnership through exchanges between scientists. So we have 300 French scientists which are based in 60 partner countries to develop this cooperation. And we have a lot of partnership and agreements with foreign universities and institutions. And I will speak in greater details on that later on. We host 1,000 foreign scientists yearly in our laboratories. And it's important figure, between 15 and 20% of our students, as a general rule on the Montpellier area in the field of agronomy. All what I presented you is about agronomy only. It's not about physics, it's not about economy, it's not about medicine and so on, only agronomy. So we have between 15 and 20% of foreign students which are hosted yearly in schools and universities. And there are a lot of Brazilian people, Brazilian students coming to Montpellier. So this is another view of that, the different types of partnerships that we have. And you see that the training agreements, this is for Europe, are a very important part. And with Latin America, we have a lot of paired diplomas, training agreements, and you see that we have connection with all the world. Okay, so this is to finish my presentation about the general overview of the different disciplines and fields that are developed in Montpellier Supagro, in Montpellier, sorry. And now I will present the Faculty of Agronomy Montpellier Supagro, to which I belong. So Montpellier Supagro is a faculty of agronomy It's called the National Institute of Higher Education in Agricultural Sciences. And we have around 1,200 students. The other students which I mentioned are in the universities. And we are developing research and education since 1842. So it's higher education. We are rather in the last levels. So we only give courses in the final year of the Bachelor of Science, just before graduation. Our main job is to teach engineers. This is why we exist, to produce engineers and to teach engineers. But we also welcome master students which work in connection with the engineers, and we also have PhD students, quite a lot of PhD students in our institution. In France, the level of engineer students is usually higher than of students from the university. This is because we are allowed in these schools to select our students, whereas in the university they are not allowed to select the students. So this is why we have usually better students. And we develop a research which is very theoretic, not really practical, but theoretic and conceptual teaching, yes, and rather than technical and practical as it is more developed here. So the subjects in which we teach are listed here. Probably it's not complete, and you see that the different levels at which they are taught. You know, mainly all are taught for engineers and for the PhD students, and some of them for the masters, and even less of them to the bachelor students. Okay, so it's post-graduation, mainly post-graduation teaching courses. We have a lot of international students, far more than what can be found in the university. We have at the master level around 30% of international students, and at the PhD level we have more than 50% of international students. And the origin of these students is from all the world, you know, and America, South and North is located here. And we have far more students from South America than from North America. The Americans do not come to Montpellier Supagro. The Americans from the United States or from Canada, they do not come. Another view up, you can see the world card. I'm sorry. So this was Europe, this was South America, this was North America, and this was Asia. And these are the list of the formal partnership for student exchanges. And you see that, of course, we have a lot of formal partnership within Europe, but the second is with Latin America. We want that our students go learning abroad, and our objective is to have 100% of our students going abroad. So this is the evolution in the last 10 years. You see, we have doubled the number of students following teaching in other countries, and this is the number of partner universities with which we collaborate. So we constantly try to increase the number of partner universities and to send our students and also to welcome students. So you are welcome, and students from the University Federal of Minas Gerais are welcome in our school. Our objective is that, as I told you, 100% of our students spend at least 3 months abroad. So we are already around since we have around 200 students in this program, we are already more than 50%. And if we add the students we do internships, we are close to 70%. And we want to go to 100%. This is why I'm all here, because we are trying to see any place in the world where it could be interesting for our students to go, and where it could be also interesting for the students from this place to come to our school. So this is the evolution of the mobility according to the destination. You see that Europe is a great part, but you see that Latin America is constantly growing, and we are obtaining more and more collaboration with Latin America. And more recently, we try to develop also collaboration with Asia. With North America, it's not possible or very difficult, and Africa, the level is very poor, and it's very difficult to send students to Africa. And the African students that we welcome, their level is very low, and it is very difficult for these students to be able to follow our courses. So there is an opportunity here to go on this development and to receive more students from your university. So we are, as I told you, involved in quite a lot of formal exchange programs, and we develop short-term exchanges programs up to 1 year long. They are called the Erasmus program. This is for Europe, Erasmus Plus program for the whole world, and we have specifically with Brazil the Brafagri and Brafitec programs. Right now, we do not have any program with the University Federal of Minas Gerais, but we could try to establish one in the future, possibly, I mean. To give you an idea of the programs that we have, the foreign students can join our programs at any level, from here to there. So this may be a little bit difficult for you because you are more involved in this part, but you could start here and then finish some courses in our school. The problem may be also that the teaching is 80-90% in French, okay, and it's needed. You need to have the B2 level. I don't know whether this classification is the same in Brazil and in Europe, but okay, you need the B2 level, otherwise you will not be able to follow the teaching. And we are developing more and more courses in English, and we are between 10 and 20% of courses which are in English, including for French people. The French people are following courses in English. So it is also possible to come for an internship, and in this case the language barrier is lower, because it's sufficient that you can exchange with your supervisor, and we have supervisors speaking Portuguese, we have supervisors speaking Spanish, so if you are more fluent in Spanish or in English, it is possible to come. And we have a lot of Brazilians coming through these ways. So I've listed here some of the programs that we have with Brazilian universities. So don't be surprised by the title. The titles are very open, so that it can enable any exchanges within the universities. All students from one university can come to our university. And it is supposed to be established between different universities. So I've listed here, for instance, at least 2 Brazilian universities and 2 or 3 French partners. I didn't list them. So these are the programs in which we are involved. So they are called Brafagri program, the 5 here, and the Brafitec, which is mentioned here. You see? And you see the different universities to which we send students. We have between 10 and 20 of our students going to this university each year, and we welcome between 10 and 20 Brazilian students in our courses for short term, between 1 month and 6 months, and sometimes 1 year. the financial support for Brazilian students is often 1 year long. So we have students coming at the different levels in our master programs. So it could be, so this is already done for year 2017 and 2018, but we can prepare the next round, and it could be possible that your university join one of these programs if the Brazilian partners are okay, or develop with us and partners other programs. So this has to be discussed if all partners are in agreement to do that. We also develop joint diplomas. That means, according to my colleague from the University of Labras explained you, students following part of their courses in their university of origin and part of their courses in another university, and in the end they have 2 diplomas from the 2 institutions. Okay? So we developed of that within Europe, but it's open to foreign students coming from abroad, from all the world. So we have 4 programs, each with at least three different European universities, and these are the global subjects. For instance, I'll show you for the plant health program. The plant health program is about protection of plants against pests. So teaching a 2-year course, master course on protection of plants. It is open for students from all over the world. You may have scholarships which cover living costs, insurance, travel, installation, and everything. So you can apply, any student can apply. It is competitive, so you have to be a good student, but it is possible. All the teaching is in English, so you need to have the B2 level. And there are some conditions here. And how is it working? Is it working like that? In the first year, you choose one of 2 universities. For instance, here, Valencia in Spain and Göttingen in Germany. The teaching is in English, okay, even in Valencia. So the first year is done in one of these 2 universities, and then the second year can be done in one of the 4 other countries. So Supagro in France, or still Göttingen in Germany, Padova in Italy, or Valencia in Spain. You see? And students change. And in the end, there is a master thesis, which is, well, common for all the students. Okay? So this is the kind of program of co-diplomation that we develop. Here are the partners, and here are the associate partners to this kind of program. So we developed this kind of joint diplomas, the Erasmus Mundus European Master of Sciences, and we also develop engineer diploma, and we are developing, historically we had one with INAT in Tunisia, which is an engineer school in Tunisia, and we have recently signed with USP ESALC in Brazil, and we will have a co-diplomation with ESALC starting in 2017. So students from ESALC, at the beginning 3 students will be involved, and we will see if it's going well. If it's going well, the 3 students from France will start 2 years in France and 2 years in ESALC, and the Brazilian student will start 2 years in ESALC and then 2 years in France. And this will be an exchange like that. We also developed that with Bambuí University in Belgium, and we aim at increasing the number of joint diplomas, and our policy is to go further in that direction. Finally, we also are involved in PhD programs. So we have 3 doctorate programs, depending on the scientific subjects which are developed in these programs. So all these programs, they largely benefit from the Agropolis Research Network. That is, the students can make their research in one of all the laboratories which are I rapidly presented you before. It is of course possible to make sandwich PhDs, no problem. We have a lot of sandwich PhDs, at least 30 or 40%. And we can also make a joint diplomation with foreign partners. That is, it's more than a sandwich, it's also a double diploma. So you have both a PhD from Montpellier Supagro and a PhD from your university of origin. So it's more than just a sandwich. Enfin, we make a difference in France between the two, okay? So it is possible to do that, and we have a lot of, enfin, we have, not a lot, but we have several of this kind of co-diplomation, okay? So this was for the presentation of the different aspects of the teaching. Then some slide to present you the environment of the school. So there is high-tech facilities to develop the teaching and the research. There is high-tech also facilities for the courses. There is also student residence accommodation. For one student out of two can be located in this residence. There is a lot of place, and the Brazilian, they come here and they organize some fests. So it's very close to the campus, and there are all facilities. For international students, we have some facilities to help them come to Montpellier. So for the scholarship funding, visa registration, and so on, organize events. And also offer French language courses all year long. And we have also a special summer school, which is a language immersion summer school, in which the students follow French classes. Of course, if you come with without knowing French, this will be not good for you. But if you have only a fair knowledge of French, only a B1 level, yes, a B1 level, you can upgrade your level and go to the B2 level through this class. There are outdoors activities with professionals in the agricultural sectors, and you have also an internship in a family farm in the south of France, so you can discover the agriculture in France. So this is a good way to get an immersion before starting really the courses which are developed in Montpellier. Okay, and of course there are all, not only working and studying, but there are quite a lot of activities which are open to the students. And I remind you there are 70,000 students in Montpellier, and a lot of opportunities to meet different peoples, to meet different cultures, and to make friends, to become friends. Okay, so this was for the presentation of the teaching activities and that we have in our schools. Now I'm going to develop some research in plant physiology that we develop in our lab. So I'm part of the biochemistry and plant molecular physiology department. This department counts around 80 permanent staff, teachers, researchers, engineers, technicians, and 72 temporary collaborators, among which 29 PhD students and postdocs. And among these, temporary collaborators, we have 31 foreigners, so we are very open to foreigners. So the general objective of the research that we develop is about uncovering and describing molecular mechanisms controlling water and mineral status of plants. It is fundamental research mainly, trying to uncover new mechanisms. Okay, so we observe something which is not explained yet and try to understand which genes, which mechanisms, which regulation explains the phenotype that we can observe. So we are not, a plant needs a light, air and carbon to make photosynthesis and to grow. We are not working on this part. We are working on the other part, the hidden part, how plants use their roots to acquire minerals and water from the soil to grow. Of course, you all know that you need nitrogen for a plant to grow and others essential minerals. So nitrogen, phosphorus, sulfate. We have 10 research groups within the department, and they study one or the other of these compounds, and also the interactions that exist between, for instance, nitrogen absorption and iron absorption, because there are links between the two. So we are working from the molecular aspect to the environmental aspects. Some projects are exclusively molecular, trying to identify genes, understand how they work, and some projects are plant physiology at the plant level, so making growing plant, analyzing their growth depending on the fertilization and the condition of growth. And we have all levels with different tools adapted to the different levels of analysis. And a lot of the programs rely to the analysis at different levels, both molecular and plant level, or including the environment. So since we work on the mineral nutrition means acquiring mineral elements from the soil into the plant. So it means transmembrane transport of ions, nitrate, potassium, magnesium, calcium, etc., sulfate. And since we study that at the molecular level in particular, the transport of ions results in an electric current. When you have a nitrate ion which is transported across a membrane, this makes a current, an electric current, because you have an ion which moves from one place to another place. And we have developed a very strong capacity in electrophysiology, which means a physiology of electric transport. Okay? So we are one of the most famous electrophysiology, we have one of the most famous electrophysiology platform in France, so this is why I present you this platform. We are working either on cells, xenopus oocytes, which are oocytes from xenopus, gourd cells, which are cells from the leaf, protoplasts, isolated protoplasts, and with different types of cells we also work on entire organs, entire roots, for instance, and we use this apparatus to inject into electrodes into these different types of cells, and we put current, we apply current on both sides of the membrane, a voltage, different voltage between the two sides of the membrane, and this induces a current, because if you have a difference in potential, you may have a transport, an electric transport. Okay? So we apply on one cell, one isolated cell, a difference of potential, of electric potential on both parts of the membrane, and we measure the current which is resulting from this application of the difference of potential. And this is the traces of current that we register. 1 nanoampere, which is very small current because it's only a few ions which transport, which go across the membrane. And here 0.5 second. And then for each level of the voltage, difference in voltage, we have a current, and we have here the characteristic of a transporter, that is the ability of the transporter to transport ion depending on the changes in electric potential. And different transporters have different capacities. Here you have 2 transporters, and they are not equivalently able to transport the ion which is considered here, which is potassium. And you may have transporters which are not able to transport in this part, but only in that part. And quite a lot of analysis of the molecular function of these proteins, these transporters. So this is what we, for instance, an example of what we are good in and what we are developing that. A lot of people are coming from all the world to visit us and to learn this technology. They send students to learn this technology and be able to apply it in their own laboratories. And so this is a hotspot for attracting students and researchers to develop specific points, specific research on this topic. We have similar tools to analyze water transport. Again, water transport across a membrane uses a protein, which is a water channel. Aquaporins, they are all. And we study these aquaporins, but we also study the whole resistance of the root system. So we study the root hydraulic conductivity. So how is it working? You put a root system into water, and all this is closed. And you put pressure here. And when you put pressure, this puts pressures on the water, and the water escapes through the plant. So the water enters in the root, and then goes up to the upper part of the plant. And the plant is cut, so that you can here measure the flux of water which comes from the solution into the upper part of the plant through the root system. And you understand that there is a resistance to this movement of water. It's not just easy. The water is not going easily from here to there. And then you can make measurements depending on the pressure that you apply here. So you put pressure here, and depending on the pressure, you have more or less water which is effluxing at this position. So you see that you can establish there is a relationship linking the flux to the pressure, and it's a linear relationship, and we can measure the slope of this relationship, and the slopes gives the hydraulic conductivity of the root. And this is linked to the tolerance to water stress. You know, when there is water stress, the plant protects itself by preventing the flux of water inside the plant so that it cannot lose too much water. Okay? So the slope here will be higher. And we try to understand what are the mechanisms which are controlling this resistance of the transport of water across the root. And we have developed quite a lot of approaches using genetics, using transcriptomics, etc., etc. And recently, a collaborator in the lab published in the very prestigious review Cell, which is the very top in biology, and to discover one of the protein, which is a kinase, which modifies the structure of a transporter and thus modifies the ability to transport water. And this can be a key to adapt the plant to water stress in specific situation. This result was only observed when there was a lot of potassium in the medium. When there was a low level of potassium, this mechanism was not working. So it is very, very specific mechanism. You understand that it has no direct practical application, but it is understanding how the plant is working, how the genes are working, and then we can expect that in 5, 10 or 20 years we can develop approaches which will use this information to make new varieties or new varieties which are more adapted to drought. Another direction which we analyze, other keywords and the main targets, we try to understand regulation networks. That is, all the mechanisms from the signal, you have drought, and then you will change the efficiency of any transporter to transport water or a mineral. So what is all the cascade of events? What protein, what intermediate compounds are between the signal and the transporter? So how many genes, how do they work with each other? Do they work 2 by 2, 3 by 3, in which situation, etc. This is what we study. For instance, here, if a ferritin, which is a protein which is involved in the storage of iron, we have identified quite a lot of different transcription factors, PIF4, PIF7, PHL1, PHL, etc., which all contribute to the level of expression of this gene. Depending on the condition, when it is cold, when it is hot, when it is when there is drought, when there is light in the night, etc. And all these transcription factors are present or absent, and they modulate the expression of the gene. And it is, we need now modeling, to try to understand exactly, to predict the level of expression of the gene, depending on all these different factors which are involved. One is responsive to the phosphorus, one to the iron, one to the day-night cycle, another one for the iron, but through a different way. So you see, it's a complex regulation network, and we try to understand this kind of network. What we also are good in, well, it's not really to see because of the light, but we analyze root development. And we have quite a lot of tools to analyze the development of a root, the speed of root growth, the number of secondary root. You see here you have a very interesting root architecture. So we analyze the architecture of the root system and see to which extent it is dependent on changes in mineral nutrition and changes in the environmental factors. Okay, adaptation to environmental factors. For instance, here you have specific roots which are induced when phosphorus is lacking, you know. And we have different markers to measure all that. And here it's difficult to see it, but we can analyze the emergence of a secondary root at this place. So very at the beginning of the emergence of this root, and to see whether this emergence is allowed or blocked, depending on the environmental conditions and which genes are responsible for that. So this is the kind of work that we develop. The model plant with which we work is mainly Arabidopsis thaliana. For those who never saw that, it is about 30 centimeters high, not more than that. 90% of our work is done on this model plant, Arabidopsis thaliana, but we also work on other model plants. Here's a pine with a mushroom, an association, a symbiosis. And we are also working on crops, wheat, rice, vine, tomato, etc. But this is more, well, this is only 10% of the total. When we study something in this species, we identify mechanisms which seems interesting. Then we go from the species to the model species, analyze precisely what is going on in the model species, and then go back here to see to which extent we can use the information and make the transfer of the information from the model species to the plant species. So we have also a lot of platforms and facilities, some of which were already presented in the previous slides. So I do not detail that. And again, we cannot see the map, which is a pity, but these are all the formal collaboration that we have between us and all the world. So we have collaboration in Europe, in Northern America, in Southern America. We have a collaborator in Professor in Brasilia. I don't wait, I've lost his name right now, and also in Asia and in Australia. So we have a lot of international collaboration, more than 30 joint publications, and several Sandwich PhD each year, and we also organize international congresses and international summer schools on our electrophysiology, for instance, abilities which are very interesting at the world level. Okay, so this was to give you a general presentation of the kind of science that we do in my department. And now I will move to two examples of subjects that we have developed in my own group. So this is a rapid presentation of the group with different permanent staff, some PhD students and master students. So we are working on adaptation of plants to metals. So you may know that there are essential micronutrients, zinc, copper, cobalt, molybdenum. These are essential for the growth of plants. But there are also toxic micronutrients, cadmium, lead, aluminium, arsenic, chromium, etc. And also essential micronutrients can become toxic when they are present in excessive concentration. And the goals of our group is to identify and characterize new mechanisms. Exactly what I told you for the group. We try to identify mechanisms, new mechanisms, involved in metal tolerance, control of metal accumulation in plants, and interaction between metal and phosphorus nutrition, because in one example that I will show you, there is a link between the two. So we mainly work on zinc, cadmium, but also on lead and iron. So I remind you, we do fundamental research. We develop some applied projects, but more in collaboration and often on short-term periods, depending on the contract that we have or not. Actually, we have a contract with a tobacco company, which is a bit long, which has 12 years long, but it's one of the seldom programs which is so long with on an applied subject. So the application of our research are in two directions. We try to improve the quality of food. So that means improving the accumulation of essential micronutrients. The idea is, for instance, in cereals, in grain, the grains are poor in iron and in zinc. And so the idea would be to enrich the concentration in iron and in zinc in the grain. This is called biofortification. We also try to improve the quality of food to limit the accumulation of toxic metals. Toxic, that is cadmium, lead, arsenic, in the logic of food security. The idea is that when you grow a plant on a polluted soil, the plant will absorb these pollutants. And they should not be present in the tissues that you eat, that people eat. So if we are able to understand the mechanisms, the transporters, the regulation which are involved in the transport of this pollutant, we can try to reduce or limit, not abolish, it's not possible right now, but to limit strongly the ability of the plant to accumulate this pollutant, which enables to maintain agriculture on soils which are degraded or not so good. And this is already practically done in Asia, in Vietnam, in China, and somehow in France you can already select natural varieties which are accumulating less of these pollutants. So the idea of our work is to understand why some varieties, some cultivars accumulate less cadmium or lead than others, and try from that to breed new varieties which would combine all the favorable alleles which in one plant, and that are more efficient for food security. The other application is about phytoremediation. So the idea is either to grow metal-tolerant plants on metal-polluted areas to fix the pollution. The idea is only to prevent erosion. When you have a polluted soil, nothing grows on this, and when there is rain or when there is wind, the pollution goes away. Okay? So there the idea is to grow specific plant on this soil to prevent erosion. So you need highly tolerant plants. And then what would be ideal would be to phyto-extract, so to extract the metals from the soil. The plant will take the metals in the soil and accumulate them in its tissues. So the metals are extracted from the soil, are in the plant, then you collect the plant and you concentrate, because you burn the plant, and you collect the metal, and then you clean the soil. This technology is not operational right now because it takes a very long time to clean efficiently the soil, but people are working on that, and we also contribute to that by trying to understand the mechanisms which are responsible for this ability to extract metals from the soil. There are already people using this as phytomining, that is plants which extract this metal from the soil, not to depollute, but to just collect the metals as a mining activity. And this is already operating, at least in Europe and maybe in other parts of the world. So in my group, we are working on metal hyper-accumulating species. So species which are extremophile species, which are able to grow on very toxic soils and which are also able to extract the metals from the soil. And we try to understand what makes these species so special. Why are they able to be tolerant and to extract this metal without being killed? We also work on crops, tobacco, wheat, lettuce, rice. And then this is to develop our biofortification programs or the food security programs. For instance, to develop varieties with reduced cadmium contents in leaves, or to analyze nutritional interactions. And we also work on Arabidopsis thaliana because a lot of the studies are done on this model plant. So what we are specifically interested in is to develop what we call open-minded approach. Open-minded approach, that is, we don't select a mechanism in advance. We just observe the nature, observe the difference between different varieties, between different species, and try to understand what is the genetic basis of this difference. Okay? So we develop diversity analysis. We compare the physiology of different varieties. And when you observe the difference, we try to understand what is the cause of this difference. And then we develop genetics approach, QTL approach. I don't know whether you are familiar with that. Cloning of the genes. We use screening of cDNA, and we also use computational biology. And this is a very modern way, and I will give you an example of what we do in that direction. And then once we have identified the genes which can explain the differences that we see between two species, then we try to understand exactly the precise role of these genes, what they do, with respect to metal tolerance and metal accumulation, what they are and to what they correspond, and I will show you an example of that later on. So, well, this is a detail. Alors, to give you a synthetic view of our current research programs, we are trying to understand what makes Arabidopsis alleri. Arabidopsis alleri, it is an extremophile species. So why is it extremophile? And we compare Arabidopsis alleri to the model plant Arabidopsis thaliana, and I will present you some results about that. We study another extremophile species, which is Hirschfeldia incana, which is tolerant to lead and which accumulates lead. This program is now stopping, but we have worked a little bit on that. I will not present you anything on that. I will present you, we have results about cadmium accumulation in tobacco, biofortification of wheat for zinc and iron, and tolerance in another extremophile species, which is Lepidium sativum. And I will present also you the computational analysis from the interaction between zinc and PI nutrition. So, one of the two examples that I will give you is about trying to understand why Arabidopsis alleri is an extremophile species. So what makes the difference between Arabidopsis alleri and Arabidopsis thaliana? Donc here you have the three species, Arabidopsis alleri, Arabidopsis thaliana, and Arabidopsis thaliana, which is a sister species of the two. These ones are sensitive to zinc, and this one is extremophile. It is able to grow on 1 molar zinc, and it can accumulate up to 5% of zinc in its dry weight. So in terms of molecular phylogeny, the history of the species, Arabidopsis alleri diverged from Arabidopsis lyrata around 0.3 million years ago, and it diverged from Arabidopsis thaliana between 5 and 13 million years ago. So this one is extremophile and the two ones are normal. So the question is, this is 300,000 years. So it is a very short time in terms of evolution. So we can expect that a very reduced number, our assumption is that only few differences explain the acquisition of the extremophile character in Arabidopsis alleri. Maybe 2, 3, 4, 5 genes, but probably not hundreds of genes, considering that there is only 0.3 million years of divergence between the two species. So, from that, we focused, a lot of groups are working on that at the world level. So, it's 1 hour. We focused on the protoplasts and the contribution of defensin to cellular zinc tolerance. And we saw that the Arabidopsis alleri protoplasts are more tolerant than the Arabidopsis lyrata protoplasts. And then we used yeast as a yeast cell model, and we expressed a cDNA library from Arabidopsis alleri. That is, we collected genes from Arabidopsis alleri and we expressed them in yeast. And we searched for yeast that were zinc tolerant. Here you have the wild type yeast, and here the yeast which express a defensin. And when expressing a defensin in yeast, we have a better growth. That is, defensin confer tolerance to zinc to yeast. So defensin are candidate genes to explain tolerance to zinc. And we validated that in the plant. Here you have the wild type plant in normal condition in the presence of zinc. So you see that the presence of zinc blocks the growth. And in the plant, which is transgenic plant expressing the defensin, we have a better growth. So, in this case, defensin proved to be important for contributing to zinc tolerance. And then, what are defensins? Defensins are small basic proteins. This is the structure. And they are involved in antifungal activity in plants. Here you have the fungus, and when you add defensin, the fungus is not growing well. And defensin also have quite a lot of other functions: inhibition of root growth, inhibition of translation, etc., and also tolerance to zinc. So the question is, do different defensin play these different roles, or so are there a specialization of function of defensin? So we identify defensin as important for zinc tolerance, and then we try to understand why, how is it working? So are specific defensin tolerant to zinc and other to other functions, or can only a single defensin play different role? So for instance, the defensin which we are working on, you see the yeast test. It enables the yeast to grow in the presence of zinc, but here it prevents the growth of Fusarium oxysporum. It has an antifungal activity. So here you have the growth of the fungus in normal condition, and when you add defensin, you see that the fungus cannot grow anymore, it is killed. But the defensin also blocks the growth of the root, and it also protects yeast against an oxidant, which is diamide. So you see that the same defensin plays a lot of different roles. So this makes the situation complex. And this leads to the concept of protein promiscuity. Unfortunately, it's not written here. That means why a protein which is so small can have so many different activities. So does it have a basic activity which results in different function depending on where and when the defensin is expressed? And this is what we are studying. I will not go into details on this slide. So what you can see here is that we've analyzed the ability to chelate zinc, would be a basic function, the ability to induce a specific unfolded protein response, which is a cellular mechanism, or the ability to produce ergosterol. These are different details which I cannot present you in details now, but which show that this protein may have different cellular functions. And we are trying to understand, but because still now we don't know how it is working. The other example that I want to show you is about the interaction between phosphorus and zinc nutrition. There is a link between the two, and for instance here, this is phosphorus absorption by the plant. When you have zinc, phosphorus is not much absorbed in the plant, and when you withdraw zinc from the medium, you have a lot of phosphorus which is entering the plant. So zinc is modulating the nutrition in phosphorus. You see? And then, okay, this is the detail. So how is it working? So I'm going, maybe you're not familiar with that. The expression of a gene is dependent from several transcription factors. Transcription factors are proteins which are inducing the expression of a gene, you see? And the idea is that you can analyze several, in the same time, different genes in different experimental conditions. So let's say that you have 1,000 genes here, and let's say 50 different conditions there. And you can measure systematically by transcriptomic approaches, genomic approaches, the level of expression of all these genes in all these conditions. Okay? And the idea of what we are doing is we are searching for transcription factors which are regulating the genes. So why is here the gene highly expressed and here poorly expressed? And does it depend from the other genes present in the organism? The idea is that one gene from the organism is depending from the expression of the other genes of the organism. And can we find out which genes are regulating the other ones? So we put a model. So it's conceptual, you see, conceptual analysis, of course, of expression. We put a model. This is a gene of interest, the level of expression. And here the different candidate genes for which we try to analyze the contribution to the expression of this gene. So how is it working? We have one gene of interest and its expression in 50 conditions. And we screen the different genes and see whether we are able to make a model which can explain the value which we have here, depending on the value that we have here, here, and here. And we change a different gene in yellow and try to find out the better association that we can make. Okay? So this can be shown like that. The measured expression of the gene is in red, And the predicted expression of the gene, depending on this formula that we are establishing, is in blue. And you see that the prediction shown here is not that bad. It's not perfect, but it's not that bad. Meaning that we are able to consider that a given set of genes is responsible for the expression of the gene of interest that we have here. And we do this is what we call computational biology. It's only data mining. You never need to see a plant, you never need to do an experiment. You just collect information from the databases and you make modeling to find out what would be the best candidate genes that would regulate your gene of interest. So it's work for the specialists in the computational sciences, in mathematics, in bioanalysis, and we work together with these people to find out new genes, new mechanisms. Yep. And the kind of results that we obtained, for instance, is that here we made this modeling. We were targeting the FO1 transporter, which is a transporter of phosphorus, and it is regulated by zinc, following the figure that I presented you before. And we identified couples of transcription factors that were explaining variation in expression of this gene. And we validated this experimentally. In the wild type, you have this response to the shorter genes in zinc. So in presence of high zinc, you have a certain level of accumulation of PI in the shoot. When you suppress zinc from the medium, you have more phosphorus in the shoot. This is what I showed you already in the previous slide. And here you have different mutants in the genes that were revealed by this computational approach. And you see that in these different mutants, we lose the response. Here we have an inverse response, the reverse. You know, we have a reduction instead of an induction. Here it's unclear, but there is not a so high increase. And here we have a very high increase, a higher increase, meaning that these different genes, the zinc finger, the BHLH, and this gene, all contribute to the response. When we inactivate the genes, we lose the response. And we could not have before any information on these genes, and we could not establish this relationship and this regulatory network that we are constructing with this work. So these two examples are to give you the level of research that we, as a general rule, develop in our lab. It is far from practical application, but it is important to understand how plants are doing. And we use this information in some projects to make applied applications to try with these genes now. We know that if we mutate them, maybe we can improve some aspects of the response, and maybe this can have a positive impact for the production. And this is what it's not, we do not do that. We collaborate with agronomists, with ecophysiologists, colleagues, which will develop the analysis in the field or in the greenhouse. And we have done our part, and then they will do their part to see whether what we have found is useful or not. And this is the way we are working in our lab. So I thank you for your attention, and I know that it was very difficult, first because of the English and also because of the size in the end, but I thank you for your attention.